My very warm welcome to the 2024 Miegunyah Distinguished Fellowship Public Lecture. For those who don't know me, my name's Sharon Lewin. I'm the Director of the Doherty Institute, which is where you are today. I know this is a public lecture, so I'm thrilled that I have many familiar faces in the audience, colleagues, friends, and of course new faces to come and hear this wonderful lecture and learn a bit more about infectious diseases, although I know that most people are now very expert in virology, epidemiology, and all sorts of things as a result of COVID-19. I'd like to start by acknowledging the traditional owners of the lands on which we are meeting, the Wurundjeri people of the Kulin Nation, and pay my respect to their elders past, present, and emerging, and welcome any Indigenous people today. I'd also like to make a very special welcome to some visitors and attendees in the audience. First of all, welcome to Deputy Premier and Minister for Education and Medical Research, the Honourable Ben Carroll. This is Minister Carroll's second Doherty Institute event for the day, so we're keeping you very busy, so thank you very much for attending. We also have the Honourable Julia Gillard in the audience, and I know that for everyone in the room, this is a huge thrill, but particularly, I think, for every woman in the room. The Honourable Julia Gillard is now Chair of the Wellcome Trust, and we're thrilled that Arup had the pull to bring you over here. We've tried before, but we needed Arup, I think, to convince you to come and visit, but we're just thrilled that you're here today. We have the Vice-Chancellor of the University of Melbourne, Duncan Maskell, and Sarah Maskell. Welcome to Duncan and Sarah. Professor Peter Doherty needs no introduction, the namesake of the Doherty Institute. Martin Meyer, the Chair of the Doherty Council. Jane Gunn, Dean of Faculty of Medicine, Dentistry and Health Science. Shelley Dolan, the CEO of Royal Melbourne Hospital. And Chris Menzies, Head of the Department of Electronic and Electrical Engineering. This Miegunyah Distinguished Lecture is a joint activity of the Doherty Institute and the Faculty of the Department of Electronic and Electrical Engineering. So we're just thrilled to co-host this together. The Miegunyah Distinguishing Visiting Fellows Program is a prestigious fellowship supported by the University of Melbourne and established by a request from Sir Russell Rimwade. These are highly competitive fellowships, and each year the University of Melbourne awards one. We've missed out on many applications before, so we're absolutely thrilled that we're able to host this year's Miegunyah Fellow. That is Professor Arup Chakraborty. Arup is an expert in engineering and infectious disease, a brilliant example of cross-disciplinary research and what it can do. He is visiting from the MIT and Reagan Institute, and Arup played a key role in the COVID-19 pandemic as a leading academic, and also has published on this area, and so is passionate about public communication. But before we get on to a full introduction on Arup, I'd like to thank a few people to making tonight possible. First of all, of course, Angus Grimwade and the Miegunyah Trust for their support of the program. Deputy Premier Ben Carroll and the Victorian State Government for their generous support of a partnership that we have with the Reagan Institute. The Reagan, similar to the Doherty, is focused on infectious diseases and immunology, and we have a visiting fellowship program together with the Reagan. So looking forward to doing much more together after Arup's visit. I'd like to also acknowledge Thas Nimalathis, a Dean of Faculty of Engineering and Information Technology, Chris Menzies, the Head of Electrical and Electronic Engineering, Professor Matt McKay. Matt's my friend and colleague who has an appointment between engineering and the Doherty Institute. And I think our experience with Matt since he came and joined us from Hong Kong a couple of years ago is a great example of what different disciplines can do together. And Matt really masterminded the invitation to Arup. I'd like to also thank Simone Robinson, Alison Broadby, and River Hugenboom from the Faculty of Engineering and IT for supporting the Doherty of the organisation of the event, as well as Eliza Copsey, Mindy Dow, and Jennifer Fagundis from the Doherty. And so I'd like to now hand over to Matt, who will give us a formal introduction to Arup. Thanks very much again for coming.
Okay, so good evening and welcome. So it's a tremendous honour to introduce the 2024 Miegunyah Distinguished Fellowship Public Lecture to be delivered by Professor Arup Chakraborty from MIT. So Arup is a highly esteemed scientist, engineer and scholar. At MIT, he holds one of the 12 institute professorships, which is the highest rank that you can get bestowed upon an MIT faculty member. He also serves as a professor of chemistry, chemical engineering, and physics. Arup served as the founding director of MIT's Institute for Medical Engineering and Sciences, creating an institute that brings together scholars from all across MIT to perform groundbreaking research to improve human health and to educate future leaders. He's a founding steering committee member of the Reagan Institute of MIT, MGH, and Harvard, and holds many other leadership and advisory roles. This includes serving on the board of governors of the Wellcome Trust, one of the world's largest charitable foundations, and as Sharon mentioned, is chaired by the Honourable Julia Gillard. Arup's research has made significant contributions and addressed diverse problems in immunology for over 20 years. His research brings together deep knowledge from the engineering, physical, and life sciences, to understand basic mechanisms underpinning how our immune system works, and to design novel vaccines against global health threats such as HIV and COVID-19. My own group members and I have had the great privilege of having Arup serve as an academic mentor and collaborator. His deep knowledge and remarkable insight have opened up a really whole new way of thinking about how engineering and data science can contribute to addressing fundamental problems in immunology and health. As a truly remarkable distinction, Arup is one of the very few individuals in the world who are members of all 3 US national academies, that is science, medicine, and engineering. He's a dedicated and passionate educator and has received no less than 7 teaching awards at MIT. So please welcome Arup Chakraborty to deliver the 2024 Miegunyah Distinguished Fellowship Public Lecture, Vaccination Through the Ages.
Well, thank you very much, Sharon and Matthew, for the kind introduction. It's really an honor to deliver this Miegunyah lecture this evening. What you will hear are the reflections of a scientist, not a historian, on how vaccination procedures have evolved through the centuries. And by way of introduction, let me say that the human story cannot be told without recounting the suffering caused by infectious diseases. Indeed, the written record is replete with references to plagues and contagion. As just a few examples, the plague of the 14th century killed the equivalent of half the European population. In the 19th century, a plague killed 10 million people in India with famous outbreaks around the world. The 20th century witnessed the famous 1918 flu pandemic, which killed at least 20 to 50 million people, many more than World War I at the same time, 15 million, including 6 million civilians. Less deadly flu pandemics followed. HIV has infected over 75 million people and likely has caused 40 million deaths. So really, COVID-19 was just a vivid reminder to us, the inhabitants of the 21st century, that infectious diseases are an existential threat. Viruses have especially wreaked havoc, particularly in the post-agrarian world. When our ancestors learned how to grow crops, they started living in large communities. So a virus that could be spread by casual contact could be transmitted to many people. Moreover, in large communities, new births and trading with neighboring communities provided a constant supply of newly susceptible people. So a virus that could be spread by casual contact and was reasonably deadly was not extinguished, as when we lived as small, isolated groups of hunter-gatherers. Then such a virus would either kill everybody in the community or everybody would become immune. Either way, the virus was done. At that time, the viruses that infected us were somewhat different. Farmers also started to domesticate animals. So the pool of viruses that used to circulate in animals could also start circulating in us. And thus it came to be that diverse viruses that could spread by casual contact and were reasonably deadly began to circulate in humans. And all of this was exacerbated by population growth, urbanization after the Industrial Revolution, and easy travel. It is interesting to compare how many we were and where we lived at the height of the Roman Empire to right after the Industrial Revolution. We went a lot of new places. Now, despite the onslaught of infectious diseases, we have survived because of human ingenuity. Very early on, our ancestors noted, and through CDC's observation in 430 BC is just one example, that those who recovered from disease could safely serve as caregivers for the sick. Such people began to be referred to as immune after the Latin word immunis, which means exempt. How we harness this observation to try and protect people from disease is best told by describing how we conquered a scourge that had plagued humanity since antiquity, and that is smallpox. The disease began with a fever, malaise, and muscle aches, and soon a rash and pustules developed in your mouth, which spread all over the body. And 30% of those infected died, and those who recovered were often left severely scarred. Now, smallpox is thought to be an ancient disease, but clear evidence for it exists in Egyptian mummies only 3,000 years ago. But as recently as the 20th century, smallpox killed 300 to 500 million people. But we don't worry about smallpox anymore. And that is because humans are ingenious. Very early, as early as 1500 AD, the Chinese were practicing a procedure to try and protect people from smallpox. Material was collected from scabs from individuals with mild disease, converted into a powder, stored for about a month, and then administered nasally through a silver tube, left for females and right for men for reasons I do not know. Someone who went through this procedure developed symptoms, usually recovered, and was usually immune. The Europeans encountered a similar procedure being used in India in the 17th century, which quickly spread to Asia and the Balkans. And the main difference was that the material taken from scabs was administered through punctures that were made in the skin and then covered with a paste made from cooked rice. Now, this procedure came to be called variolation and was brought to the West by a remarkable woman, Mary Wortley Montague. She and her family had suffered a lot from smallpox, and she observed variolation being practiced as the wife of the British ambassador to Constantinople. And so she had her son variolated by Charles Maitland, the British doctor there. Upon her return to England in 1721, there was a very serious epidemic in London, and Montague had her daughter variolated as well. Then a child of the Princess of Wales, Caroline, fell ill, turned out not to be smallpox, but nonetheless the princess was worried. Montague and others promoted variolation to her, and thus began perhaps the first organized and highly publicized clinical trial. 6 condemned prisoners in Newgate Prison were told that they would be set free if they underwent this procedure. So in August of 1721, they were variolated. 5 developed symptoms and recovered. The 6th, that turned out, had had smallpox the year before. And when one of those who recovered was sent to care for a very sick child, they were able to do this without falling sick. But the princess was worried about safety for her children. So 5 orphans were variolated in March of 1722. And again, no harm. You can see that our ethical and statistical standards for clinical trials have evolved somewhat. Anyway, the princess was satisfied and so had her 2 daughters variolated, thus temporarily popularizing this procedure in the upper echelons of British society. But really, variolation was a very traumatic and dangerous procedure. It often caused serious or lethal illness. Sometimes it sparked localized smallpox outbreaks. And so variolation was not practiced very much. Enter Edward Jenner. So Jenner was born in May of 1749 in Berkeley, England, and after finishing his studies in London, he returned to Berkeley in 1773 to practice medicine. Now, cowpox was a harmless disease for humans that was transmitted to milkmaids from the nipples of infected cows when they milked them. Now, it is said that a milkmaid told Jenner that she did not respond to variolation because she had had cowpox before. And he heard similar other stories, so he started to research cowpox. And it took a while because cowpox outbreaks were not frequent. But ultimately, Jenner was ready to try an experiment. On May 14, 1796, the variolation procedure was used on James Phipps, but with material from cowpox scabs taken from Sarah Nelms, who was infected by a cow called Blossom. We know all this about this because it was a momentous event in history. 2 months later, Phipps was variolated, but this time with smallpox, and he did not develop symptoms. Now, the British Royal Society refused to publish Jenner's initial paper on this subject in their venerable journal, the Philosophical Transactions, saying that he didn't have enough data and he would embarrass himself. So Jenner had this published by the private firm of Sampson Low and presumably made some money. Now, Jenner's finding was really a paradigm shift because what he said was that you didn't have to immunize people with material taken from a deadly infection. Rather, you could immunize people with a related infection, cowpox, that was harmless. And so it would be safe, and it proved to be effective. So in 1958, using Jenner's procedure, the WHO led an international vaccine campaign that led to the eradication of smallpox in 1979. The last known case was that of Janet Parker, who was a medical photographer who was accidentally infected in a lab. Today, 2 vials remain, one in the US and one in Russia. And some wonder why, because smallpox could probably be synthesized in a laboratory for research purposes as a related cowpox virus has. Now, Jenner is rightly celebrated for ending a disease that had plagued us forever. But vaccination was not without opposition, sometimes very strong. One example was in the US. The Boston and New York epidemics in early 20th century were really devastating. And Massachusetts allowed towns to impose mandatory immunization orders. In 1902, the town where I work, Cambridge, passed such an order. And the penalty for not complying was a $5 fine, which is about $180 today. Now, Henrik Jacobsen, a Swedish immigrant, refused to be immunized and refused to have his family immunized, saying that it would make him sick. And he didn't pay the fine and sued Massachusetts. So in 1905, the United States Supreme Court ruled in favor of Massachusetts, saying that Jacobsen exercising his rights would endanger the community. Now, vaccination is an understandably scary procedure. You are asked to inject something in yourself which is related to a deadly pathogen. Now, I'm not a social scientist or a politician, but I think scientists and public health officials should clearly and transparently lay out the benefits of vaccination for the individual, for the community, and the low statistical risk of modern vaccination procedures, as well as what we do not know, so that people can make hopefully informed decisions based on trusted news. Now, remarkably, Jenner did not know that microbes cause diseases, nor did he know much about anything about the immune system. So the discovery that infectious diseases are caused by microscopic organisms required the development of high-powered microscopes, such as those developed by Van Leeuwenhoek, who was a Dutch businessman and amateur inventor. His microscopes were the most powerful of the time. They were only about 2 inches long. But with them, he started observing details of the stinger of a bee, mold growing on bread, and he observed little objects running around in lake water. He called them animalcules. He made meticulous drawings of these things and sent it to the Royal Society, which initially did not believe this animalcules business until an investigative body came and determined that it was true. Of course, his animalcules were really bacteria, which can cause terrible diseases, but Leeuwenhoek did not know that. This realization came in the 19th century with the work of Robert Koch in Germany and Louis Pasteur in France, who were bitter rivals. Now, Koch was a precocious child, and he invented the way to isolate bacteria from people and growing them in the laboratory, which was called, a process was called growing in culture. He also proposed the rules for identifying the causative agent of diseases. First, the microorganism must be present in every instance of the human disease. It must be grown in culture, and this microorganism, if it was injected into an animal, must produce exactly the same disease as in the human. And a microorganism taken out of this diseased animal must be identical to the one that was taken from the human. These Koch's postulates are used to identify the causative agent of bacterial infections and choose treatments. Pasteur was an indifferent student as a youth, but he made some important discoveries. For example, he discovered that molecules could be left or right-handed. So if you take your left hand and make a mirror image of it, you'll see your right hand. And you won't be able to superimpose these two. That is the definition of handedness. Now, molecules can be that way too. For example, if you take this molecule and take a mirror image of it, it'll look like that. And you won't be able to superimpose these because this guy will end up there and this will end up here. Molecules that have a handedness are called chiral. Sugar is a chiral molecule, and our digestive system can only process the right-handed form of sugar. Of course, he invented pasteurization. But all of these major accomplishments have been overshadowed by his transformative work on vaccination. So Pasteur injected the bacterium that causes chicken cholera into chickens, and they did not fall fatally sick. He then realized that the culture had spoiled. So then he recultured it properly, and re-injected the same chickens, and they did not fall sick at all. Pasteur is said to have noted that in the field of observation, chance favors the prepared mind. Well, his mind was definitely prepared because he instantly understood that a weakened or attenuated form of the pathogen in the spoiled culture could confer protection. Of course, without knowing anything about microbes, our ancestors were trying the same thing with variolation. This business of storing and powdering and drying was basically an attempt to do exactly that. Now, Pasteur advanced this concept and developed a bunch of animal vaccines, but he was dying to do something for humans. So rabies is an infection of the nervous system, and you usually get it from dog bites or bat bites. It's caused by a virus, so Pasteur could not use Koch's postulates to grow the virus because viruses are tiny, a 1/1000th of diameter of a hair. He's said to have been fearless and extracted saliva from a rabid dog. And then with Emil Roux, he attenuated the virus by passing it multiple times through rabbits. And the idea was that by doing so, the virus would adapt to rabbits and so it would become less deadly for humans and dogs. He then took their spinal cords and dried them and took material from it. In 1885, Joseph Meister was badly mauled by a rabid dog, and Pasteur vaccinated him with this before symptoms took over, and he was saved. Pasteur became famous, and he loved it. He was quite the character. Apparently, when he was director of scientific studies at the Pasteur, at the École Normale in Paris, when students refused to eat the mutton stew, he decreed that the stew would be served every Monday. Now, by the way, Pasteur really did not know why these procedures were protective because he really didn't know much about our immune system. Today, we know that our immune system is made up of two inextricably linked parts. One part is called the innate immune system, and a computer scientist would call the innate immune system a classifier because the cells of the immune system have antennae on their surface or receptors, some of which can recognize molecules that are usually on the surface of all viruses. Others can recognize the ones that are usually on the surfaces of diverse bacteria, and so on. So it classifies the kind of infection you have, viral, bacterial, parasitic, etc. And then accordingly, it secretes products that slow the pathogen's growth and generally create an inhospitable environment for the pathogen. Some of this makes you unhappy too. For example, you get a fever. But the innate immune system does another very important thing. It activates the second part of our immune system, which is the adaptive immune system, and instructs it about the type of response required based on the kind of infection. But the innate immune system is a blunt instrument. You can only tell it's a virus, it's a bacteria, not which virus, which bacteria. The adaptive immune system is remarkable because it enables us to mount pathogen-specific responses against a diverse and evolving world of microbes. To roughly see how this works, consider infection with a virus. When a virus infects a host cell, it hijacks its machinery, enabling the synthesis of new viral proteins and the assembly of new virus particles that can go on to infect other cells. Now, B cells are important players in adaptive immunity. We each have billions of B cells, and most have a receptor on their surface that is distinct from the receptor on the surface of another B cell. This, by the way, was first understood by Burnett here in Australia. If the receptor on a particular B cell can bind sufficiently strongly to the surface proteins of this virus, it gets activated, it starts to multiply, and then a Darwinian evolutionary process takes place in a short time that I will describe a little bit later, that ultimately leads to secretion of soluble and even higher affinity forms of these receptors, and that's what we call an antibody. These antibodies can bind specifically and strongly to the spike or the surface proteins of this particular virus and then dispose of it using the innate immune system. But antibodies principally do battle with virus particles that are free in your blood or space between your cells. Now, some of these viral proteins are chopped up into short fragments called peptides and displayed on the surface of these host cells with host proteins called MHC proteins, and that's the molecular flag of an infected cell. Professor Doherty made some seminal discoveries about these molecules, the MHC molecules, and thus met the King of Sweden formally in an evening in December. Now, T cells are also key players in adaptive immunity. Again, we have billions of T cells, and most have a distinct receptor on their surface. And if the receptor on this particular T cell can bind sufficiently strongly to this viral peptide, it gets activated, starts to proliferate, and then can coordinate an immune response in a number of different ways. For example, certain kinds of T cells called CTLs are such that if an activated CTL sees another cell displaying the same peptide that originally activated it, it will secrete products that will kill this infected cell and the virus particles that it harbors. Now, after you've cleared an infection, most of the B cells and T cells that multiplied in response to this particular infection die. But a few remain as so-called memory cells that can mount robust and rapid responses upon reinfection with the same pathogen. This pathogen-specific immunological memory is the basis for vaccination. With a vaccine, you try to induce memory B cells, T cells, and antibodies that are specific for the bug against which you wish to protect the host. So now you see how these 19th century vaccines worked. There were two kinds, those that contained the live attenuated pathogen and those that contained the inactivated or killed pathogen. Injecting these elicited memory B cells, T cells, and antibodies that were specific for the proteins of the pathogen, which then conferred protection upon natural infection. You also see now why this killing had to be done just right, because if you mangled these proteins too much, then the response they would generate would not be specific for the proteins of the live pathogen. Now, these 19th century technologies are still used, for example, in your annual flu shot. And they were used very fruitfully to combat polio. Also, an ancient virus that sometimes can infect the nervous system and cause paralysis. It's a waterborne disease carried by infected fecal matter. And in the 20th century, polio outbreaks happened with regularity. Counterintuitively, mostly in countries that had better sanitation. And if someone's interested later, I can describe why that was so. Jonas Salk, a New Yorker, designed an inactivated vaccine. He killed the poliovirus just right with formaldehyde. The clinical trial, which was a logistical tour de force, was successful. People lined up to take Salk's vaccine, and he became a US national hero. Fellow New Yorker and rival Albert Sabin was developing a live attenuated vaccine. By the time he was ready to try it in humans, the March of Dimes and the US, the March of Dimes, the principal sponsor of Salk's vaccine, they had a vaccine. They weren't interested anymore. It is remarkable that at the height of the Cold War, the first major clinical trial and manufacturing of an American vaccine was conducted in the Soviet Union. Sabin's vaccine was approved for US in 1961 and soon replaced Salk's vaccine because it had many advantages. One was Salk's vaccine had to be administered intramuscularly, whereas Sabin's vaccine, you just put a dollop on a sugar cube and you ate it. This is how I was vaccinated. The other was that Salk's vaccine produced a kind of antibody called IgG antibodies that protected the blood and the nervous system so you didn't get paralyzed, but it didn't protect the intestinal surface from being infected. So the person could still pass it to others through fecal matter. Sabin's vaccine produced IgG antibodies, but also another kind of antibody called IgA, which protects surfaces, and so the intestine was protected from infection. By the way, there's a major efforts underway to develop vaccines that will induce IgA-type antibodies to protect mucosal surfaces from respiratory viruses. Now, the 20th century saw new types of vaccines too, because of advances in biology and biotechnology. In the 1950s, we learned that all the information about all our proteins, which carry out all our functions, is encoded in DNA. And this DNA, the information contained in the DNA, is first transcribed into a related molecule called RNA, which is then translated into the sequence of amino acids that constitute a protein. In the 1970s, recombinant DNA technology came along, which was that you could isolate a piece of DNA encoding information about a desired protein. Then you could take a piece of DNA out of a bacteria, swap out a piece of it with this one, stick it back into the cell, and now this cell became a factory for making the desired protein. So vaccine developers thought, since the immune system targets the proteins of a pathogen, why not just inject proteins? That would be safe because there would be no pathogen. They're easy to manufacture using recombinant DNA technology. They're killing the virus just right, and so on and so on. But the trouble is, if you just take proteins and inject it into an animal, nothing happens because it doesn't activate your innate immune system, and therefore it doesn't activate the adaptive immune system. So with these sorts of so-called subunit vaccines, you also have to add other components called adjuvants, which activate the innate immune system. A very successful example of this sort of vaccine is the one that protects against human papillomavirus, which causes cervical cancer. The 21st century saw new kinds of vaccines during the COVID-19 pandemic, where instead of injecting the proteins of the pathogen, information regarding those proteins was injected. In the AstraZeneca and J&J vaccines, DNA corresponding to the spike protein of the SARS-CoV-2 virus, which causes the COVID-19 disease, was inserted into the genome of an engineered adenovirus, which could not replicate and therefore could not cause disease, but it could still enter our cells, and then our cells' machinery converted this DNA into the spike protein of the virus and thus elicited an immune response. The BioNTech and Moderna vaccines encoded information about the spike protein as RNA and encapsulated it in a lipid nanoparticle. Lipids are also components of our own cells. And these nanoparticles are designed to be preferentially eaten up by our cells. Then, of course, the RNA is translated to the protein by our own machinery, and immune response results, which confers protection, and the RNA is quickly degraded. Now, these vaccines have proven to be remarkably safe and effective. So, are we done? Well, we don't have effective vaccines against highly mutable viruses. HIV is a prominent example. We don't have a universal vaccine against the seasonal flu either. So every year we guess which strains might evolve and design a new vaccine. And SARS-CoV-2 seems to be in a similar place now. What is required to confront these scourges better are universal or variant-proof vaccines. A part of my work aims to address fundamental questions which, if answered, would aid the pursuit of this quest. In the remaining few minutes, I'm going to try to give you a high-level view of some of this work. I will not describe details. And that's because, well, let me tell you a story. One evening, Princess Julia of the Principality of Gilad imprisoned two of her subjects. One of them was a theoretical and computational scientist like myself, and the other was a normal person. Princess Julia informed the prisoners the next morning that they would both be executed, but they were granted one last wish. The theorist jumped up and said he had discovered this beautiful equation the night before, and he wanted that a symposium be held where the great mathematicians of the land would come and debate the nuances of this equation. The princess said, fine. Turned to the other person who said, my last wish is that I want to be executed before this symposium takes place. So no details today. Now, seriously, let's frame the challenge. This is a schematic depiction of the spike of HIV. It has some regions that don't vary much from one strain to another. For example, this region shown in yellow, which has to bind to human receptors in order to infect our cells. But these conserved regions are surrounded by highly variable regions shown in red and shielded by these sugars shown in blue. Now, the influenza spike also has a little conserved bit here that binds to human receptors, but it has another big conserved region in the stem of the spike that is necessary for entering cells. New antibodies have been isolated from some patients that, at least in the lab, can neutralize diverse strains of either HIV or flu. These so-called broadly neutralizing antibodies, or BNAbs, bind to regions that contain these conserved bits. But upon natural infection or typical vaccination, the vast preponderance of antibodies that are produced target all these variable bits. So BNAbs are very rare. Nonetheless, their existence is proof that the human immune system can generate such antibodies, and so raises the tantalizing possibility that properly designed vaccination procedures could induce them efficiently in diverse people. Now, antibodies are produced by a Darwinian evolutionary process called affinity maturation. It really is a portrait of how life itself evolves. As I mentioned, if the receptor on a particular B cell can bind sufficiently strongly to the spike proteins of a virus, it gets activated, and then these activated naive B cells, so-called naive B cells, they concede structures called germinal centers, or GCs, in your lymph nodes. And here, the B cells start to multiply, but also mutations are introduced at a very rapid rate into the antigen binding region of their receptors. These B cells with mutated receptors then interact with the antigen displayed on nearby cells. If the receptor on this particular B cell can bind sufficiently strongly to the antigen, when the B cell pulls on it, it eats it, chops it into little fragments, these peptides that I told you about, displays them on its surface with Peter Doherty's molecule, and then the B cells that have eaten antigen and display these peptide fragments compete with each other to interact with T cells, and a productive interaction leads to survival. Otherwise, the B cell dies. So you can see that a B cell with a receptor that binds more strongly to the antigen is more likely to eat more antigen, and stochastically more likely to display more of these peptides on its surface, and so stochastically more likely to win the competition with its peers. Now, some of these positively selected B cells, they exit as memory B cells or plasma cells. And these plasma cells secrete soluble forms of the receptor or antibodies. But the vast majority are recycled for further rounds of mutation and selection, and with the bad ones die. So you see now why, as time ensues after vaccination or natural infection, the antibodies get more potent because they've gone through more rounds of mutation and selection. Now, upon natural infection or typical vaccination procedures, the evolutionary paths predominantly lead to antibodies that bind all these variable bits. So the evolutionary paths that lead to BNAB-like animals, if you like, are very rare. Our challenge is to design vaccination protocols that make these rare evolutionary paths to BNABs more likely. And what we have at our disposal are variant antigens that share the conserved regions, but the variable bits are different from each other. We predicted through our computational studies that sequentially immunizing with these variant antigens, that is, give one, then wait, give the second one, wait, give the next one, is going to be more effective at eliciting BNAbs than if you gave a cocktail of all these antigens at the same time. And many experiments in mice have now shown the efficacy of sequential immunization for making progress towards BNAbs. We also predicted that there is an optimal difference between the variable bits of the sequentially administered antigens to maximize the chance of BNAb evolution. And signatures of this has been seen in humans who, HIV-infected humans who naturally make BNAbs, and in our own work with Dan Lingwood on flu antigens in mice, where if the sequentially administered spikes were too similar, there were no stem-targeting BNAbs. If they were too different, nothing happened. But if they were sufficiently different, stem-targeting BNAbs emerged. But our excitement about these sequential procedures has been given some pause because of the following reason. Michel Nuzensweig and Bill Schief did a beautiful experiment in engineered mice where they gave sequentially variant HIV antigens and saw progress towards making BNAbs. When a very similar experiment was done by Pamela Bjorkman and Michel's lab, but this time not in engineered mice, but in an outbred population of monkeys, you saw evolution towards BNAbs, but as the number of shots increased, the response got diluted with new responses. So to understand the mechanistic origin of this observation in primates and perhaps learn how to go around it, we decided to learn from the biggest source of primate data, COVID vaccination of humans. And with these two MIT students, and in close collaboration with Michel Nuzensweig's lab at the Rockefeller University, we asked, why do 3 shots of the COVID vaccines confer protection against a highly mutated variant like Omicron, when 2 shots do not, when every shot contains the same sequence, the original Wuhan strain? So these studies have taught us that antibodies generated in the previous shots influence the evolutionary processes that ensue in subsequent shots in very interesting ways. And these feedback loops from pre-existing antibodies ultimately lead to the evolution of these novel responses, which were originally subdominant responses. In the case of COVID, this was beneficial for us because it protected us from Omicron. In the case of that HIV monkey experiment, it was not beneficial because it led to new responses that diluted the one that we wanted. More generally, what we are learning now is that which type of antigen is there, when it's there, how much is there, and what's the past history determine the outcome of these processes. And so what we are trying to do now is to optimize the design of variant antigens and the temporal pattern in which they are administered and how much is administered in order to optimize desired antibody responses. And we are trying to do this by bringing together physics-based computations, approaches from machine learning, learning from humans, and tests in animal models in an iterative fashion. I look forward to describing some of our recent results following these paradigm in my 2 technical lectures at Melbourne. I had hoped that I would be able to provide you with a glimpse of one today, but my time is up as I had anticipated. So let me just end by saying following. In the end there, I've tried to give you a little flavor of the kind of work that I do with my wonderful lab members and collaborators. But really, we are a small slice of a large community of scientists in academia and industry who are working toward these variant-proof vaccines. Success will be another step forward in our eternal war with infectious diseases and will help create a more pandemic-resilient world. Thank you for listening. Thank you. I'd be happy to answer questions, so Sharon's going to take over.
Well, thank you, Arup, for an absolutely spectacular lecture. You said to me earlier that I wasn't going to learn anything, but I learned a lot. And I think it's a special art to be able to explain something so complex with brilliantly simple slides, a few fascinating stories, which I had not known about, Blossom the cow and others. And then to end with the complexity of the work that you do. And having worked in HIV for a very long time and knowing a lot about the story of broadly neutralizing antibodies, you explained it just brilliantly. However, I did want to add one last thing to the story about broadly neutralizing antibodies, and that was by knowing what you described with HIV and influenza, that led to us being able to develop therapeutics for COVID using broadly neutralizing antibodies actually at remarkable speed.
Yep.
So all of this incredibly detailed knowledge that we get from some viruses and the investment that we make in science for those viruses really does pay off, and we saw that in COVID. And I'm sure you'd agree with me that those principles were lived out.
More than principles. I mean, Sharon's absolutely right. I mean, these DNA vaccines and RNA vaccines did not suddenly develop, but it's not suddenly developed for COVID-19. They've been under development for 10 years, some for HIV actually, and other diseases. So I think keeping on doing the sort of work that it sets us up for success when bad things happen.
Exactly. So thank you very much for a superb public lecture. I'm sure this is the one that you sweated the most about, as opposed to talking about your area of specialty. And I do think we have some time for a few questions. So any questions for Arup Chakraborty? And no question is a stupid question. Yes, is that Tim? Yeah.
Yeah, thank you very much. Tim Stinier from the Doherty Institute. My question is, could you comment a little bit more on your last slide, where you showed us a Venn diagram of where you hope to be and your iterative cycle of learning? Where are you in that learning? Because I feel like perhaps there are some black boxes there we still need to uncover.
No, for sure. I mean, in my own case, typically, when we started 24 years ago working in immunology, this was the iterative cycle, just between these 2 boxes. Then it became all 3. And I think learning from human data is critically important for this stuff. But now we also, with the advances in machine learning and large language models and so on and so forth, it is very interesting to integrate various types of computation together, because it is my belief that one can learn more by bringing these together than either could learn by themselves. Now, one problem that does exist in immunology more than virology is that it's difficult to generate truly high throughput data for immune systems, but it's happening now in single cell RNA-seq and so on, but people just make UMAPs, as you know. I mean, it's okay, but how much do we learn? So, I think there's a real opportunity of bringing these two kinds of computation together with these things. And I do have some examples to show in the 2 talks in April.
Other questions for Arup? While people are thinking, I'll ask you a question, Arup, and I'll ask you a non-scientific question or a non-biological question, because our biggest challenge in COVID was not developing the vaccines, it was getting people to take them. So tell us a little bit about vaccine hesitancy over the ages and what's worked and what hasn't, and why don't people trust the science?
Well, you do understand that if I really knew the answer to this question, the big problem would be solved. But look, I can tell you only about what the reasons are in the country where I live. And but I suspect there are similarities. I already mentioned it's a scary thing to ask people to do. But if I tell you that your chance of getting serious, and by the way, if you go look, vaccine adverse events are cataloged by the CDC in at least two databases, the Vaccine Adverse Event Reporting site and the Vaccine Safety Data Link. If you go and look, between 2006 and 2013, for every 1 million doses of a vaccine, there was 1 adverse event. And usually they were mild, but there were some very dangerous ones. But if you tell someone that your statistical chance of being hurt by the vaccine is less than when you drive a car, it's not very real for people. They drive a car, nothing happens to them. They don't take vaccines every day. So part of it is understanding risk, that is not just a number, is a challenge for people. And then the other part is that the few events you hear about, like the J&J vaccine, caused Guillain-Barré syndrome, caused clotting for some people, few in a million, but they died. They affect you much more seriously than a statistical number that the risk is not so high. And then, of course, the one place that I do not exist is social media. It generates a lot of misinformation. And also think about this, all this stuff about autism being caused by vaccines, one retracted paper. That's the origin of it. So it's a complex issue. And I don't know how exactly to handle, but it was quite intense and it's getting more intense.
That's not easy at all, at all. Other questions at all for Arup? Yes.
Thanks for a fantastic lecture. So welcome back at the Doherty Institute as well.
Thank you.
I thought when you were talking about vaccination through the ages, how all these infectious diseases have shaped human evolution, has vaccination has a positive selective pressure as well then?
I don't know. I do not know. I should not, let me pick a different example that's not about MHC because Doherty is sitting here, but we do know that past infections, our genome is a fossil record for past retroviral infections. I can't remember now, but a few % of our genome are ancient retroviral infections that still remain in our genome, passed down from across the generations. Just like someone who's infected with HIV, which is a retrovirus, their genome has this forever, right? All infected cells. So it certainly has influenced parts of our genome. I don't know if they have influenced human evolution in other ways than simply that we have not all perished. Many people are alive. Yeah.
Yes.
Thanks, Sharon. So Ben Cowie from the Doherty in the Department of Health Victoria. And thank you for a fantastic lecture.
Thank you.
You've talked about the fact that we now have human data, particularly through the COVID-19 pandemic, to a degree that we've never had before, both in terms of viral evolution and also evolution of our immune responses, both to vaccination and to recurrent infections. One of the issues that we've had, despite the amazing rapidity of vaccine development and deployment, has been the evolution of the virus getting away from our vaccine platforms and the need to continuously update those formulations. With the machine learning and physics-based computations that you described, do you see that in the future, for a similar pandemic, we could predict what evolution the virus may go through in successive waves so that we actually start designing vaccines for viral sort of convergent evolution that is yet to happen in a year or two, so we've got them ready to go?
I think it will happen for several reasons. One is this is where you can collect high throughput data. You can sequence the hell out of these things, right? And collect all over the world and just get sequences. In fact, Matthew has tried to do some of this, and he collaborated with us in at least one paper for our work on HIV in this field. Okay, where it's not how does it evolve in individuals. And right now there are companies that have been formed. I should say it's there in my declarations. I'm involved with one of them, which is trying to do precisely that. And you don't have to get necessarily a vaccine that works perfectly for all these different strains. If you get one that is good enough for various possible strains that may emerge, that's enough of a safety net while you design a perfect vaccine for what's going on now. And in one of the things I will show in my technical talk at the Doherty is some work that we've done with Pamela Bjorkman, which at least in mice seems to do that. But as you know, mice lie and monkeys don't always tell the truth. But at least in monkeys. So I think it will happen.
Okay, well, I know we're a bit over time. So before I ask you to join me in thanking Arup for a fantastic lecture, Arup, we have a few gifts for you. A beautiful bottle. I assume it's Australian. I hope it's Victorian, more importantly. It is indeed. Yarra Valley Chardonnay.
Thank you very much. And I think Matthew told you about my taste in wine.
I hope you enjoy it.
I shall.
And well, a book from Peter Doherty, who knows a thing or two about viruses, the immune system and pandemics. This is Peter's book around the pandemic in Australia, particularly Melbourne. It's called An Insider's Guide, An Insider's Plague Year. I'm not sure whether you should have the wine before or after the book, but I know you'll enjoy it and learn a bit about what happened in Australia. Thank you very much.
Thanks very much.